Hello, my name is Katz Amburn. I'm here to talk to you about my poster for evaluating genomic assemblies and annotations using publicly available platforms. I am an adjunct researcher in the Department of Biochemistry and Microbiology, and I'm interested in how you go from having small DNA pieces you get from Illumina sequencing to an assembled and annotated genome. There are several publicly available platforms that have programs to assemble and annotate genomes. These include the four listed here: Galaxy, KBase, BV, BRC, and Proxy, as well as an annotator only, which is RAST. My objective was to evaluate the differences in the assembly and annotation of the same set of Illumina reads. I know that many platforms have the same programs, but do they give the same results, and what's best for me to use to submit to GenBank? My methods: we created an Illumina library from a clinical Serratia marcescens strain. I submitted the reads to each platform and assembled by all programs and compared the results. I picked the best, Assembly, and then I submitted it to each platform and the RAS server and annotated again by each program and compared the results. This is the first table of data I want to show you. On the left side are the platform I used on that column. The next column over is the assembly program. The next is the number of big pieces that there were once assembled. Then the genome length, which was fairly comparable to the type strain for the bacterial species. Then the largest contiguous fragment, or contig, and an N50, which is a measure of the contiguity. You want to have as few contigs as possible, and the largest contig and the largest N50 as possible. I found that the unicycler bold on Galaxy gave me that. In the second table, you see what happened when I put that unicycler bold assembly into all of the different platforms and ran it through the annotation programs. You find that this table is now separated by the annotation program, and the annotation program along with the platform that it was present in. And that's so that you can see that even with the same annotation program in a different platform, you get somewhat different results. You get a fairly consistent but different number of coding sequences, are ribosomal RNAs and transfer RNAs. You find that the number of unidentified proteins, which is in this case a sum of the hypothetical, the DUF, domain of unknown function, the UPF, unknown protein function, the UC, the uncharacterized proteins, the PP, in other words, putative proteins, they all add up, but they're very different numbers even when using the same annotation program. The variability in annotations is also illustrated in this table, where I now have columns indicating the annotation program and the platform that it was done in, and you can see the different kinds of features that are discussed or listed in each of these annotations, as well as the differences in the annotations even between the same annotating program in a different platform. My take-home conclusions really are: there are considerable differences in assembly and annotation, and I used this process to determine the best platform and program for assembly and annotation for this strain, and I will continue to do it for the other strains that I have to analyze. I came out with some thoughts to recommend to other people. The first is, if you're assembling reads and annotating for submission to something like GenBank, do both processes in more than one program and platform and pick the best. And if you're doing a comparative study looking at the same genomic features in different strains of organisms, download the FOSTA-DNA file. Run that same FOSTA-DNA file through a new annotation using the same annotation program for all of them so that when you do compare, you are looking at apples and apples. That concludes my presentation. I hope to see you at my poster. Thank you very much.